All right, guys, this is Objective 3B. Objective 3B is going to cover genetics. I'm going to talk about the difference between Mendelian genetics versus non-Mendelian genetics. Now, Gregor Mendel is known as the father of genetics, and he is known for his studies on pea plants. And so he came up with three different laws of genetics, the law of dominance, the law of segregation, and the law of independent assortment. The other thing that is essential for Mendelian genetics is there must only be two different alleles for a trait, and the gene for the trait must be located on an autosome. And an autosome is a chromosome that does not determine gender. If it does not follow these rules, whether it's only two alleles for the trait or it's on an autosome, then that's considered non-Mendelian or advanced genetics. Now, when we look at the different laws that Gregor Mendel set forth, the first one is the law of dominance. The law of dominance basically states that one allele can mask another allele. So in this case, we're going to use red and blue, where we're going to say red is dominant to blue. So an organism that has two copies of the red allele is going to be homozygous. The reason why you have two copies of the allele is because you have two biological parents. It's for sexual reproduction. There's going to be two copies of the allele, and so there's two red alleles, then that phenotype, or what it looks like, is going to be red. So my genotype is homozygous dominant, and my phenotype is going to be red. Now here, notice the phenotype is also red, but my genotype is not homozygous dominant. My genotype is heterozygous. I have two different versions of this allele. I have the red allele, but I also have the blue allele. The blue allele is represented by lowercase r because it is, in this case, recessive. Because in complete dominance, one allele is recessive to the other one, meaning it can't be masked. So even though the phenotype shows red, the genotype has a blue allele there. It's just been hidden. So this will be a carrier for the blue allele. This is a heterozygous genotype. Now the next one, notice that the only time I see the blue phenotype is when it is homozygous. Because when there is a recessive allele, the only way to actually see the recessive phenotype is if it is homozygous. You cannot be heterozygous recessive. This pattern of inheritance is known as complete dominance, okay? And this is Mendelian genetics, where we talk about complete dominance. Now, the next law, the next one of Mendel's laws is the law of segregation. And this law basically tells us that when an organism has two alleles for a trait, when the gametes are being formed during meiosis, those two alleles will separate from each other. So in this case, I have a heterozygous mom, so the mother is heterozygous for her alleles, but when her gametes are formed, half of them will get the dominant allele and the other half will get the recessive allele. So her two alleles separate from each other during gamete formation. The last law is the law of independent assortment, and we can examine the law of independent assortment by looking at a dihybrid cross or higher. Because the law of independent assortment states that their chance of inheriting one allele does not affect your chance of getting another one. So you getting one trait does not influence your chances of getting a different trait. This is true whenever genes are far apart on a chromosome so that they can be separated during crossing over, or they are located on different chromosomes so that the chance of getting one does not affect the chance of getting another one. Now, when we examine the law of independent assortment, we have to look at the phenotypes more so than the genotypes. So in this case, the mother is purple and smooth, but the father is pink and dented. So their offspring inherited the purple from the mother, but just because they got the purple from the mother does not mean that they're going to also get the smooth. So they got the purple from the mother, but the dented from the father. So them inheriting one trait did not affect them inheriting the other trait. So these are the different things that Mendel observed when he looked at his pea plants, and that was because each one of the plants that he looked at for the traits he examined, there was only two different versions of that particular trait. Now, when we look at non-Mendelian genetics, this is a type of genetics that does not hold to Mendel's laws, okay? So with non-Mendelian genetics, there is either not going to be a recessive allele, there's going to be more than two copies of the allele is going to be located on a sex chromosome, and usually we look at the ones on the X chromosome because there's more genes on the X chromosome, or the genes are linked to each other. Now, linked genes do not adhere to the law of independent assortment. Linked genes are genes that are located on the same chromosome, and they're so close together on that chromosome that when crossing over occurs, they do not separate from each other. So your chance of getting one is directly influenced by your chance of getting the other one. Our example in humans is going to be red hair and freckles. Now let's look at what happens when you don't have a recessive allele. So if there is no truly recessive allele for the versions of the genes that I have, I can either have incomplete dominance or I can have codominance, okay? Incomplete dominance is when the heterozygote is going to show neither phenotype of a homozygous trait. And the codominance, because co means both, the heterozygote is going to show both phenotypes of the homozygous trait. So notice I only care about what's happening with the heterozygote here with incomplete dominance and codominance, because if it's homozygous, it's going to be what it is. So again, with the incomplete dominance, there is no recessive allele, and the heterozygote shows a blend or an intermediate of the homozygous traits. So we're still using red and blue. So I can be homozygous for red. I have two red alleles, homozygous for red, so I'm going to be red. If there's two blue alleles, I'm going to be homozygous for blue, so I'm going to have two blue alleles. But notice when I have one red and one blue, my phenotype, I get a third phenotype that is neither red or blue. It is instead a blend of red and blue, which is purple, okay? So my heterozygote is going to be a blend in the phenotypes. Also notice I did not use a lowercase letter because there is no recessive alleles, and lowercase letters indicate a recessive allele. Now when I get over to codominance, my heterozygote should show both traits of the homozygous. So again, there is no recessive allele because both alleles are indeed dominant. So both dominant, codominance, that's what that means. And the heterozygote is going to show that because a dominant trait is going to be expressed. Again, with the homozygotes, if I have two red alleles, I'm going to show red. If I have two blue alleles, I'm going to show blue. But in this case, with the heterozygote, if there's one red and one blue, then my phenotype is going to be red and blue. I'm going to see both of those homozygous traits being expressed. Now, the next type of non-Mendelian genetics deals with multiple alleles. Remember, with Mendel, you can only have two versions of the gene, so only two alleles. But if there's more than two alleles, then that is considered non-Mendelian genetics. And also, anytime there's more than two alleles, there's a possibility that I could have more than one pattern of inheritance. Our patterns of inheritance are either complete dominance, where one allele can be hidden, incomplete dominance, where the heterozygote is a blend, because there's no recessive allele, or codominance, where if it's heterozygous, I'm going to see both traits. Now, our example for multiple alleles that we're going to use is human blood types. There's three different alleles for human blood type. There's the allele A, allele B, and the allele O. Notice that both A and B have a capital letter because A and B are both dominant. But O is a lowercase letter because the allele for O is recessive. So in human blood types, I have three alleles and two patterns of inheritance. I have codominance between A and B, but I have complete dominance if O is involved because O is recessive to both A and B. So with those three alleles, that also gives me four different blood types. So now I have four genotypes instead of four phenotypes instead of three. Type A, I can be homozygous or heterozygous. If I have two A alleles, I'm homozygous for A. But if I have an A allele and an O allele, I'm heterozygous for A because O is recessive to A. If I'm type B, I can be homozygous or heterozygous. I can have two B alleles and be homozygous for B, but if I have one B and one O, then I am heterozygous for B because O is recessive to B. But if I get one A allele and one B allele, this is codominant, so my blood type will be AB. And the only way to be type O is to have two recessive alleles because O is recessive. So if I'm type O, I will have two O alleles. I can only be homozygous for type O. Now our last example of non-Mendelian genetics is going to come from sex-linked traits. If a gene is located on a sex chromosome, it is considered a sex-linked trait. Now sex-linked traits tend to affect males more than females because males are hemizygous, meaning they have two sex chromosomes, but one of them is an X and one of them is a Y. Females are XX. They have two X chromosomes. Genetically, you are considered a male if you have a Y chromosome, okay? So genetic females are XX and genetic males are XY. So let's do an example where we're going to talk about hemophilia, which is a sex-linked recessive trait. Now, I can have any pattern of inheritance with this, okay? Because I'm only looking at where the gene is located with the genders, with the X's and the Y's. But this can be recessive, it can be inherited dominantly, it can be inherited co-dominantly. So my patterns of inheritance can all apply here. The key is, when we look at these, we look at traits that are located on the X chromosome. So we say if it's sex-linked, it's usually X-linked, which means my Y chromosome is only there to determine gender. Anytime you have a sex-linked trait and you're trying to evaluate its inheritance pattern, you want to always do a gender box first. Before you add your traits, you do a gender box. One of the parents is female, so they're going to be XX. The other parent is male, so they're going to be XY. When I do my gender box, that's going to give me two females, so two daughters and two sons. This is important because gender matters when it is sex-linked. So you have to do your gender box first. Now, let's look at a cross between a female who is a carrier for hemophilia and a male who has hemophilia. Whenever your traits are sex-linked, males cannot be carriers. Only females can be carriers because if it's sex-linked, it's X-linked. And females have two X's, but males only have one. So they either get it or they don't. So in this case, I have a female who is a carrier. And since hemophilia is sex-linked recessive, that tells me the gene for hemophilia is a lowercase h. So a capital H means I don't have hemophilia. My female is a carrier, so on her X, I'm going to put one big H and one lowercase h because carrier means heterozygous. Now my male has hemophilia, which means he only has the little h on his X. I put nothing on the Y because if it's sex-linked, it is X-linked. Now I'm going to do my Punnett squares if I would normally do one. I'm going to put my big H on one X, a little h on the other one. Here, I have a little h and a little h. Here, I only have the big H on this X because there's nothing on the Y. And here I'm going to have the little h on that X because there is nothing on the Y. If it's sex-linked, it is X-linked. Now, multiple questions can be asked here. You can be asked how many females have it, how many females are carriers, how many males have it, how many males don't have it, how many females don't have it, how many children in all. So if I'm talking about the children, I'm talking about all four. But if I'm specifically listing gender, I'm only talking about those particular boxes. So if I say how many females, my answer can only be 0, 50, or 100 because there's only two females. But if I say how many children, there's 4 children, so then it can be 0, 25, 50, 75, or 100. So you have to pay attention to gender when it comes to a sex-linked trait. All right, I'm going to do another genetics video on genetic technology, but as far as Mendelian and Non-Mendelian genetics, that is it. Toodles.